my name is Kimberly Garcia. I'm a McNair Scholar at the University of Texas at San Antonio. And today we will be exploring the role of general amino acid permeases in mediating Candida albicans sensitivity to the calcineurin inhibitor drug FK506. So all of this research came from a mycology lab. So show of hands real quick, who here knows what mycology is? Good. So for those of you who didn't raise your hand, mycology is a study of fungi. Fungi are eukaryotic organisms, and this includes yeast, molds, and mushrooms. Now, fungi are not to be confused with bacteria. They're often associated together because we can't see them and they cause us harm. But they're actually in two different domains of life. Fungi belong to eukarya and are eukaryotes just like us, meaning they have a lot of more genetic similarities to humans than they do with bacteria. You probably don't think about fungi much, except whenever you're seeing if your food went bad the next day in the fridge, but we use them in all sorts of fun, interesting ways, such as yeast for bread, making beer, antibiotics. We use penicillin has come from a mold, and they've made appearances in popular media, such as the cordyceps and the Last of Us that don't actually hurt people. While these are all real cool fun guys, they're not all great guys. So fungal pathogens are responsible for 13 million infections and 1.5 million deaths a year, according to the World Health Organization. This is real concerning. The leading cause of this problem is Candida albicans. This fungal pathogen has been labeled a critical priority. It's the organism of interest in my lab. It is the most clinically prevalent fungal pathogen, and it lives amongst your normal microbiota commensally, meaning it doesn't normally cause you harm, but it can. It can cause an illness called candidiasis. There's two types of candidiasis. The first is mucosal. It's the most common. This can be oral thrush of the mouth or vaginal yeast infections. Both of these are uncomfortable conditions, but can be treated easily with antifungals. The second is the more serious of the two. Systemic candidiasis can cause a blood infection, which can lead to death. This is the more serious one because probably 70% of you in the room already have Candida inside of you or on you. But not to be alarmed, because this only really happens to immunocompromised people. These are the ones who are at the highest risk of getting sick. Some conditions that make you immunocompromised are cancer, AIDS, surgery, and even diabetes. These can all weaken your immune system and leave you susceptible to fungal infections. And this is why Candida albicans is a great opportunistic pathogen, because as I said, it's already on or in you right now, so whenever your immune system drops, that's the best time for it to infect you. So right now, there are only 4 classes of antifungals that currently exist. That's terrifying. So the most popular of these is the azoles. Azoles are great, but fungi have shown resistance to the azoles, and whether that be inherent resistance or misuse or overuse of the drugs. Now, the other 3 are not widely available or safe for humans to use. So this really emphasizes the need for new novel treatment. But that can be really challenging. That can be expensive and cost a lot of time. So what we can do is repurpose already existing drugs. So the drug I listed there is what I'm working with. It's FK506. It is an immunosuppressant drug currently being used for transplant recipients, so their bodies don't reject the transplant. It's known to impact the virulence of fungal pathogens, despite it not originally being intended for an antifungal. And most importantly, it's a calcineurin inhibitor. In case you don't know, calcineurin is a phosphatase. It's widely conserved amongst eukaryotic species, including pathogenic fungi. So you might be saying, if it impacts the virulence, why aren't we currently using it? That's because, as I mentioned earlier, fungi are eukaryotes just like us. That makes it very challenging when finding drug targets because our systems are so similar, and as you can see here on the figure on the left, I have the calcineurin signaling pathway of a human next to fungi. They look incredibly similar, and from a distance, it may look like I just changed the colors. So if we were to currently target calcineurin right now, we would be hurting our human host because that would drop our immune system, and as I just told you, that would make you immunocompromised and you'd be susceptible to fungal infections. We don't want that. So there's ongoing research right now to find analogs of FK506 That way it'll only target fungi calcineurin. But as fungi do, they build resistance to drugs. So they have shown resistance to FK506, which is why we're looking at this pathway to identify the mechanisms of resistance, where they are, and how we can prevent them from happening. So in a lab at UTSA, in the lab of Dr. Lee, he and Dr. Vilenki discovered a novel pathway of resistance in the fungal pathogen Mucor cercinoloides. It's a zygomyces and a black mold. Normally in a laboratory setting, FK506 is used on Mucor to keep it in its less virulent yeast form, which is smooth. However, Dr. Vilenki observed that some of the isolates were resistant to FK506 because they would continue filamenting. Upon further investigation, he found that they all had a mutation on the gene BICA, which encodes a general amino acid permease, a GAP. Because of this, he did some further investigation as to why this was happening, and they found this pathway. So normally what happens is when calcineurin is inhibited in Mucor, there's an upregulation of cyclic AMP, which activates protein kinase A, which triggers a signaling cascade. However, the mutation on the gene BICA disrupted the signaling pathway, which then caused the isolates to become resistant and bypass the inhibition that was being caused by FK506. With this, he wondered, does this pathway exist in other fungal pathogens? So there was some investigation into basidiomyces, Cryptococcus uniformans. But they saw that there is some involvement, there's still ongoing research on that. There is amino acid permease involvement, and we do know right now that when we inhibit calcineurin, Cryptococcus cannot grow at 37 degrees Celsius, which is important because that's our internal body temperature. If it can't grow at that, it can infect you or kill you. So this led us to question, does this pathway also exist in the more clinically relevant fungal pathogen, Candida albicans? And if it does, what are the GAP's roles and involvement in this pathway? So we know that when calcineurin is inhibited in Candida albicans, it can't grow in serum. This is important because, as I mentioned, that nasty blood infection can't occur. Serum mimics the conditions of the blood. If it can't grow in serum, can't grow in your blood, it can't kill you. So this is all for a pan-fungal drug. This would be the ultimate goal, to design a pan-fungal drug that is effective against multiple fungal species. That's why I'm telling you about these other two fungal pathogens that weren't in the title of my presentation, because it's effective in these three that are from different fungal phyla, they're different species. This would be a great common target for this pan-fungal drug. So going into this project, we had a few objectives. We needed to determine if this pathway exists in Candida albicans. We needed to understand the calcineurin signaling pathway to identify resistance mechanisms and explore the roles of the GAPs and their roles. So before I explain, you're gonna see this key a couple times. So lucky for us, Candida albicans is a very well-characterized fungus. This was good because we were able to obtain mutations of the fungus, and it had 6 knockouts. So Candida has 6 GAPs. We were able to individually knock them all out. Now, what we were looking for is resistance. So we'll be looking at the R column. Everything I'm gonna show you, we're gonna look at it vertically in columns. So we're looking for that R, looking for something that grows further down the plate than the control that'll always be on the left. Unfortunately, we didn't see any resistance when we did the knockouts. So as you can see, they all look pretty similar to our control, SC5314, which is a clinical isolate of Candida albicans that is standard in our lab. Well, this means 2 things. The pathway doesn't exist, or there's functional redundancy amongst these GAPs. So in order to find out if that's true, we did the opposite. So we hypothesized that the overexpression of the GAPs in Candida albicans should increase the sensitivity to calcineurin inhibitor drug FK506 when grown in serum at 30 degrees. In order to do this, we used a plasmid that our lab constructed. It has a conditional tetracycline promoter, so we were able to turn it off with the addition of doxycycline. This would tell us if any of the phenotypes that we're observing are real. So we chose GAP1 and 5 to be cloned and transformed into Candida albicans. And then this is them plated out. We use these transformants to set up our spotting assay. That was the biggest part of this experiment. So the first day we set them up in liquid YPD. and we put them in a shaking incubator overnight, 30 degrees. That is the optimal temperature for growing Candida. The next day, we measured out the OD and standardized it to 1. We then added our FK506 drug and serum, put them back in the incubator to see how they grew. The following day, we did a serial dilution in a 96 well plate and spotted them out onto a YPD agar plate, growing them again overnight at 30. This is that lovely key again. This time we're not looking at the R column, we're gonna be looking at the S column. We wanna see if anything grows less than our control, that'll be on the left, so we don't want them to grow as far down the plate. This time, we did see something different. We saw there was some sensitivity in gap 5. So you can see 5A and 5D specifically don't grow as far down as our control there on the left. So we know that the overexpression of gap 5 does inhibit more. It's more inhibited than our wild type. So in order to test that what we were seeing was a real phenotype, we added doxycycline, which is for that conditional promoter that we had. This turned off the overexpression of gap 5. And from here, if I would direct you to 5C and 5D, you can tell the ones with the doxycycline grew as normal, but the ones without, with the overexpression, didn't grow. This tells us that this was the result of the overexpression. What we were seeing was real. So, in conclusion, we suspect that this pathway probably does exist in Candida albicans, which is good for us because we can continue on. And we know that the GAP5 does play some role in the sensitivity pathway, but GAP1 does not. And this means there's partial functional redundancy of the gaps since they don't do the same thing. For the future, we'd like to see if any of the other 6 gaps, if we overexpress them, if they would have the same effect on the sensitivity. We'd also like to test the PKA activity. This was something they did in Mucor and Crypto, so we like to find that commonality for that pan-fungal drug. And we'd also like to see if it exists in other Candida species, such as Candida glabrata, which is a haploid, unlike albicans, which is a diploid. This makes it easier to identify any mutations. And we've done some preliminary work here, and we've seen that 138, it's a clinical isolate, does show some sensitivity, so we'd love to do more work with that. And I'd just like to acknowledge my PI, Dr. Saville, and our master's student, Cassidy Graham, who's amazing, and the McNair Scholars Program. Thank you very much, and I'll take any questions.